Chapter 4, A Tour of the Cell. Humans versus bacteria. What are antibiotics? Antibiotics are drugs or medicines that kill bacteria. So the objective of antibiotic treatment is to knock out invading bacteria while causing no or minimal damage to the normal or healthy cells in the human body. And this is possible because antibiotics are very specific in their action. That means they are very precise. They bind to structural constituents found only in bacterial cells. They will not bind to human cells and harm them. Now, some examples of antibiotics include penicillin, which was the first antibiotic to be discovered. Additionally, there is erythromycin, streptomycin, there is ciprofloxacin, and all of these antibiotics bind to different parts of the bacterial cells. The advent of antibiotics was so successful and the incidence of bacterial diseases went down so much that in the early 1900s, it was hypothesized that all bacterial infectious disease would be wiped out from the surface of Earth. Unfortunately, this did not happen. Why? Cell is the basic unit of life, and all living things are made up of cells. Cells come from division of pre-existing cells. So living organisms can either be very simple and made up of a single cell, then they are known as unicellular organisms, for example, prokaryotes or protists, or living organisms could be more complicated and they can be made up of many cells. In that case, they are known as multicellular, for example, plants, animals, and fungi. Photographs taken with microscopes are called micrographs. Light microscopes can magnify up to about 1000-fold, while electron microscopes use beams of electrons rather than light and can show objects about 100 times smaller than light microscopes. There are two major categories of cells: prokaryotic cells and eukaryotic cells. The prokaryotic cells are smaller, simpler, they lack membrane-bound organelles, and they are found in bacteria and archaea. While the eukaryotic cells are larger, they are more complex, they have membrane-bound organelles, and they are found in protists, plants, fungi, and animals. This table shows a comparative analysis of prokaryote versus eukaryote. This figure shows you the detailed structure of a prokaryotic cell. A eukaryotic cell can be further classified into a plant cell or an animal cell, and the structures are shown here on this slide.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are two types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracts. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
Let's begin with an overview of a eukaryotic cell. Membranes partition eukaryotic cells into functional compartments. The largest organelle is usually the nucleus. Other organelles are located in the cytoplasm, the region outside the nucleus and within the plasma membrane. Membrane structure. The plasma membrane, or the cell membrane, is the outermost boundary of the living cell, which separates the cell from its non-living surroundings. The plasma membrane and other membranes of a cell are mainly made up of two layers of special types of fats called phospholipids. Phospho means a phosphate group and lipid is a fat. And these phospholipids are arranged into a two-layer sheet, and this two-layer sheet is known as phospholipid bilayer. An important property of the plasma membrane is that it is a fluid mosaic. Fluid because molecules can move freely past one another, and mosaic because of the wide variety of proteins in the membrane. The figure here shows the structure of a plasma membrane, particularly the phospholipid bilayer. Each phospholipid is made up of a water-loving hydrophilic head and a water-hating hydrophobic tail. So you see the peculiar arrangement of the phospholipids with the hydrophilic heads on the outside of the cell as well as towards the cytoplasm, while the hydrophobic tails are in the center. There are two main types of proteins in the plasma membrane. The first one is superficially attached to the cell membrane that can easily be ripped off. Those are known as peripheral proteins, while those proteins that are very tightly integrated into the phospholipid bilayer are known as integral proteins. In addition to these proteins, plasma membranes also contain cholesterol, which provide fluidity to the plasma membrane. The walls that encase plant cells support plants against the pull of gravity and also prevent cells from absorbing too much water. Animal cells are coated by a sticky extracellular matrix. In recent years, antibiotic resistance has become a huge problem. This happens because When bacterial colonies are exposed to antibiotics, natural selection would favor those bacteria that can survive and reproduce. So, over the course of time, more and more bacteria get resistant. So, scientists are always trying to produce new antibiotics. This is an ongoing effort. In one such experiment, scientists, they took plastic devices that were compartmentalized and they grew individual soil bacteria type into these compartments. That means each plastic device had different bacteria growing in the different compartments, and the partitions kept the bacteria separate from each other. These devices were then buried in the original soil and bacterial colonies were allowed to grow further, and then finally each bacteria type was tested for its ability to kill Staphylococcus aureus, which can cause deadly MRSA, which is methicillin resistant S. aureus infection and tuberculosis. This is an illustration of the experiment. After screening about 10,000 types of different bacterial strains, scientists found out that the bacterium which was most effective in killing Staphylococcus aureus was a new species that produced a new type of antibiotic called Teixobactin. Now we are starting to learn about the different cellular organelles. The first one is nucleus. Nucleus is the main control center of the cell. Why? Because it has the genetic material, your chromatin. The chromatin is made up of DNA and proteins. DNA is like the king of the cell that issues all orders, while proteins are the workers. They do all of the cell. The nucleus is kept separate from the cytoplasm due to the presence of a nuclear envelope, okay, which is also known as the nuclear membrane. And the nuclear envelope has pores, openings that allow things to move in and move out of the nucleus. In addition to the genetic material chromatin inside the nucleus, we also see some densely packed structures which stain very deeply. Those are known as nucleoli. Singular is nucleolus. This is important because this is where ribosomes are made. Ribosomes, after being made in the nucleolus, inside the nucleus, they move out of the nucleus, they go into the cytoplasm, and then they are the protein making factories. This figure illustrates the structure of nucleus. What is the relationship between DNA, chromatin and chromosome? The genetic material present inside your cell when it is long and thin, when the cell is not dividing, it's called chromatin. And when the cell is about to divide, the same genetic material becomes short and thick. It's known as condensation, and then it is known as a chromosome. So you can call it chromatin or chromosome. These are made up of DNA and protein. The DNA is long thread-like, it's like a string, and the proteins would be circular, bead-like. So the DNA or the thread-like material is wound around the bead-like protein. Ribosomes are the protein making factories of the cell. This is where protein synthesis occurs. Ribosomes themselves are made inside the nucleus, within the nucleolus. Then they move out through the nuclear pores, and they could either swim freely in the cytoplasm, then they are called free ribosomes, or they could get attached onto the surface of the endoplasmic reticulum, which is another organelle, and then they become fixed ribosomes. The figure shows a model of a ribosome in process of synthesizing a protein. These are endoplasmic reticulum on which ribosomes are attached. How does DNA, your genetic material, direct the production of proteins, the workers? DNA contains all of the information that is required, right? But DNA is the king, so it stays inside the nucleus. It makes a photocopy of itself, and the photocopy is known as RNA, or more specifically messenger RNA. So mRNA is made inside the nucleus, and then it moves out of the nucleus through the nuclear pore. It goes into the cytoplasm, and then the information which is contained in the mRNA is read or is used by the ribosomes. The ribosomes also need the help of some other kind of RNAs which are present in the cytoplasm called transfer RNAs, and then all of these constituents together help in making proteins. This figure depicts how the DNA is giving the information to the mRNA, and the mRNA is moving out into the cytoplasm, and with the help of ribosomes it is forming the purple colored protein molecule. So what do you think is the major theme illustrated here by this action? A hint is information flow. There is an endomembrane system inside the cell. Okay, and these are mainly responsible for manufacturing and distributing various products of the cell. The endomembrane system consists of, it starts with the nuclear membrane or the nuclear envelope, then it continues as the endoplasmic reticulum, then next one is the Golgi apparatus, lysosomes, and finally vacuoles. Now these organelles are all membranous, they have membranes around them, and these are either physically connected or they talk to each other by means of sac-like structures called vesicles. The endoplasmic reticulum consists of membrane-enclosed tubes and sacs within the cytoplasm. It is usually connected to the nuclear envelope and it serves as the main manufacturing facility in a cell. The figure shows endoplasmic reticulum. Rough endoplasmic reticulum is named because of the ribosomes attached to its surface, and its function is to make membrane and secretory proteins. Sometimes the products made inside endoplasmic reticulum are packaged in sac-like structures which are called transport vesicles, and then transport vesicles might take these products to the Golgi apparatus. This figure shows you how ER is manufacturing and packaging secretory proteins into transport vesicles that get bud off from the ER. The smooth ER lacks the ribosomes on its surface, and the functions include lipid synthesis and detoxification. The Golgi apparatus is a stack of flattened vesicles. It receives the ER products and then packages them further in secretory vesicles and then sends them to their final destination, which could be another organelle inside the same cell, or it could be exported from the cell. This shows the function of Golgi apparatus. Comment on the major theme illustrated by this action. Hint: interactions within biological systems. A lysosome is a sac-like structure which is membrane enclosed, and it is filled with very strong hydrolytic or digestive enzymes. Plant cells usually do not contain lysosomes. Lysosomes aid in digestion and recycling or dismantling of things within the cell. Lysosomes can destroy anything that is undesirable in the cell. It could be a harmful bacteria, it could be an organelle that has become old, or sometimes if the whole cell needs to be destroyed, then a signal goes to all the lysosomes in a cell and they all rupture, releasing their strong enzymes, and the entire cell is destroyed, it is digested. That is known as autolysis. This figure depicts two functions of lysosomes.
Like secretory proteins and some other proteins, proteins destined for lysosomes are made on ribosomes bound to the rough ER and move through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself.
Vacuoles include the contractile vacuoles that expel water from certain freshwater protists and the large multifunctional central vacuoles of plant cells. This figure shows the two types of vacuoles. The plant cell central vacuole may absorb water, it may store organic nutrients, or it may contain pigments to attract pollinating insects. This figure helps you to review the endomembrane system. The two organelles that serve as the cellular power stations are the chloroplasts and the mitochondria. Chloroplasts. Chloroplasts are the organelles that perform photosynthesis in plants. Photosynthesis is the process of conversion of light energy of the sun into chemical energy of sugar and other organic molecules. This entire world is running on the energy which is provided by photosynthesis. This figure shows what the inside of a chloroplast looks like. Mitochondria are found in all eukaryotic cells, and this is where the energy producing reaction occurs. Mitochondria use oxygen and glucose or other macromolecules to produce ATPs, adenosine triphosphates, which is molecular currency. This is what is used by living things for energy, and carbon dioxide may be produced as a waste product. This is what you'll see if you cut open a mitochondrion. Very surprisingly, other than the nucleus, the two other organelles that contain their own DNA are chloroplasts and mitochondria. Their DNA is a little bit different, and it is suggested that long time ago chloroplasts and mitochondria were independent cells and they could survive on their own. Now by some chance, some accident, these got inside the body of other cells, and then that symbiotic relationship was beneficial to both, and they just continued to survive inside other cells. This slide just describes what we've already discussed in the previous slide. So guess what is the major theme illustrated by this action? Cytoskeleton means skeleton of the cell. Now because the cell itself is so small, so what does the cytoskeleton look like? It looks like thread-like. Okay, it's a thread-like structure. These are protein fibers which are extend or present throughout the cytoplasm, and their function is to give support to the cells and maintain the shape of the cells. There are three types of cytoskeleton. The thinnest ones are called microfilaments. The intermediate ones, which are not very thin, not too thick, are called intermediate filaments, and the thickest ones are called microtubules, which are hollow tubes of protein. This figure shows microtubules. Some examples of where you would find microtubules are in cellular structures like flagella and cilia. What is a flagella? A flagellum is a tail-like structure, and you can find this in the sperm tail. So that the tail of the sperm is made up of flagellum, and that is the only cell inside human body that contains the flagellum. Now cilia on the other hand are shorter than the flagellum, and they are hair-like structures which usually move in a wave-like fashion, and inside the human body you would find cilia in the respiratory tract, in the windpipe. The cilia are shorter and more in number than the flagella, and the cilia move together in a coordinated back and forth motion. Both cilia and flagella can help small organisms like protists to move through water. Some examples of flagella and cilia.
Paramecium moves by coordinated beating of its many short cilia. The cilia form longitudinal rows along the entire body of the cell and into the oral groove. The beating action of the cilia moves the paramecium forward and backward. It also produces a slow rotation of the cell as it moves through the water. The cilia in the oral groove propel food particles into the cell mouth, where the food can be ingested by phagocytosis. Credit: Graham R. Kent and Rebecca L. Turner.
So defects in cilia and flagella can cause respiratory problems or male infertility. As per the rules of evolution, there is struggle for existence and survival of the fittest, which means that if a disease is present persistently, then this can provide a new basis for measuring or researching those individuals who are best adapted for survival in the local environment. An example is that of Bangladeshis who have lived for long in an environment and they frequently suffer from cholera, which is caused by bacteria. So it is expected that natural selection would favor those individuals who have some resistance to these cholera causing bacteria. Data revealed that people from Bangladesh indeed have mutations in several genes that confer or provide an increased resistance to cholera. Now this could be good news, and pharmaceutical companies can in future exploit these proteins produced by the identified mutations to create new antibiotics.